So the important functions of membranes are to allow for transport. We spoke about our fluid compartments, the intracellular fluid compartment, the extracellular fluid compartment, and it's really important for these two compartments to exchange materials and to communicate with each other. Now the transport is selective, so the membrane will determine what can go across and what cannot. This is what we mean when we say semi-permeable. And this helps to keep separate substances in each of these compartments. We'll go on to see that, for example, there's more sodium in the extracellular fluid than there is potassium. There's more potassium in the intracellular fluid than there is sodium. And that is able to be brought about because of the semi-permeable nature of the membrane. So some of the things that can transport across are oxygen and nutrients, which can go into the cell. What does oxygen do when it goes into the cell? Where does oxygen go? What is the function of oxygen? It's going to go right to the mitochondria. It's going to go right across the inner mitochondrial membrane, and it's going to be the final electron acceptor, right? Remember that from the oxidative phosphorylation that we talked about last week, okay? So oxygen needs to get into the cell, so do other nutrients, such as glucose and so on. And then the waste products, which are byproducts like carbon dioxide, ammonia, need to leave the cell and be transported out into the blood. So selectively permeable means that it allows some things through and it restricts others. And it does so based upon charge or size or polarity. So nonpolar molecules are going to be able to cross the membrane, and we just explained why. Because they can exist in that non-aqueous middle of the phospholipid bilayer. So because nonpolar molecules are usually fats or things that can exist well in water, they can go through the phospholipid bilayer because they can exist in that non-aqueous center. Some of the examples we'll talk about later on, like steroid hormones. Oxygen can also go right through the lipid bilayer, carbon dioxide as well, and our fatty acids are some examples of things that can go through. Now, ions, which are charged, and polar molecules such as water cannot go through the membrane. Glucose, protein, sodium, potassium, calcium, these are examples of particles and molecules that are either charged or polar, or sometimes too large, like proteins, to go straight through the membrane. Okay? So this is what we mean when we talk about selective permeability. Okay. So we're going to move on to objective 2, which is to talk about the role of a few forces, the chemical force, the electrical force, and then we're going to put them together and look at the electrochemical driving force. And this is how substances move across the membrane. We'll also distinguish between passive transport and active transport and the energetic needs of these two processes. So passive transport is a spontaneous process. It's a downhill movement. It needs no energy, but it yields energy. So it frees up energy, liberates energy for other molecules to move, but it does not itself require energy. It's spontaneous in this way, and it happens according to gradients. So if we have a high concentration of a substance on one side of a membrane, a semi-permeable membrane, and we have low concentration of substances on the other side of that semi-permeable membrane, we have the passive movement from high to low concentration. Active transport, on the other hand, is non-spontaneous. It needs energy. It cannot happen in the absence of some kind of energetic liberation. So usually passive transport donates or liberates energy for active transport. So this is an uphill movement, and so we need some type of energy. So in terms of exergonic and endergonic, passive transport would be exergonic, it's going to liberate energy. Active transport would be endergonic, it's going to require energy. Okay. So we're going to start looking at these forces separately, and then eventually we're going to put them together. So both forces combined are the electrochemical force, but separately it's the chemical and the electrical. Okay. So we'll start by looking at the chemical driving force. First of all, this is governed by the concentration gradient. And the concentration gradient is a fancy way of saying the difference in concentration across the membrane. So again, looking at that example of a semi-permeable membrane that separates two fluid compartments, if we have a higher solute concentration on one side and a lower solute concentration on the other side, automatically we have a concentration gradient because of the difference in particle concentration. Now this gradient creates a force that pushes these molecules from the area where they are present in abundance on the high concentration over to the area where they're lower in number, which is the lower concentration. And so the force that pushes those particles is the force that we're describing here. That's the chemical driving force. Okay, so let's illustrate that here. So the chemical driving force is created by the movement of particles down their concentration gradient. You can also describe it as a force that is moving particles from high to low concentration. So let's look at a scenario here. A cell. We're going to look at particle A, and this can be substituted for an ion such as maybe sodium, which is high in concentration outside. But for now, we'll just call it particle A. So particle A is in higher concentration outside of the cell. And lower concentration inside. And then particle B is at lower concentration outside the cell and a much higher concentration inside. Okay. So automatically we have a concentration gradient for both of these particles. What direction is particle A going to want to move? Into the cell. Perfect. So particle A is going to want to get into the cell according to its concentration gradient. And then particle B is going to want to go in what direction? Out of the cell. Yeah, perfect. Out of the cell. So again, according to its concentration gradient, particle B is moving from high concentration to low concentration. Okay. Let's point out that this is a semi-permeable membrane. So if you were to graphically represent this here, from the inside, particle B is moving out according to its concentration gradient. But from the outside, particle A is moving in according to its concentration gradient. Okay, so they're both moving down their concentration gradients in opposite directions. So this is the chemical driving force.